Hello, Assalamu alaikum. Here is Mujibullah Chauhan. Today I discuss about equipments, instruments, glasswares and non-glasswares used in microbiology laboratory. Hot air oven. Hot air oven is used for sterilization. It sterilizes the glasswares and non-glasswares. Drying oven. Drying oven is used to dry the samples as quickly as possible by removing moisture from the chamber. Autoclave. It is also used for sterilization and also known as steam sterilizer. It steams under the pressure to kill harmful bacteria, virus and fungi. Incubator. It is used for to grow cell or microbial culture. Deep fridge. It is used for the storage of samples, specimens, vaccines and medicines at a specific temperature. Electronic top and balance. Used to measure the samples. Electronic analytical balance. It is used for the small amount of samples like in milligrams. Shaking incubator. Used for the cell culturing. The instrument incorporates oxygen and evenly distribute nutrients through the culture media. Gel electrophoresis is used to separate mixture of DNA, RNA or protein according to molecular size. Radioimmunoassay machine used to measure concentration of substance, usually measuring antigen concentration. Real-time PCR machine is used for measuring DNA using PCR. Distilled water plant. It is plant used for purification of water by removing chemicals, heavy metals, microorganisms and sediments. pH meter used to measure hydrogen ions activity in a solution. Microscope used to observe small objects which I can't see on naked eye. Electron microscope used for obtaining high resolution image of biological and non-biological specimen. Cubic colony counter. Counting bacteria colonies that are presented against a dark background. PCR Thermocycler are instruments used to amplify DNA and RNA sample by polymerase chain reaction. Shaking water bath used in testing for food and beverage materials as well as bacterial culture. Hot plate used for heating object. Refrigerated centrifuge used to separate fluid, gas or liquid based on density. Electronic cell counter. The name implies are used for counting live or dead cells in the samples. Spectrophotometer measure the substance of concentration such as proteins, DNA, RNA, growth of bacterial cells and enzymatic reaction. Magnetic stirrer used to make stir bar immerse in a liquid, quickly spins or stirring or mixing a solution. Automatic bacterial identification system. The name implies it is used automatically identify the bacteria in a very short time. Sonicator is widely used in the laboratory to disperse non-tube into polymer matrix. Vortex Mixer are one of the primary technologies for mixing laboratory samples in test tube, well plate or flask. Laminar flow chamber or cabinet are used in laboratories for contaminative sensitive process like plant tissue culture. Electronic wet balance used to measure the samples. Microscopic slide used to hold object for examination under microscope. Test tube used to hold, mix or heat the samples. Petri dishes. The dish is used to culture cell by providing storage space and preventing from the getting contaminated. Inoculation loop and needles are devices used for inoculation and stir dilation, stir sampling, transfer and microbiological samples specimen. Pipettes and tips are used generally moving samples amount of liquid. Maconkey Agar is used for isolation of gram-negative enteric bacteria. Test tube holder used for hold the test tube. Salmonella, Shigella agar or SS agar. It is used as a selective and differential medium for the isolation of Salmonella and some Shigella species from the clinical and non-clinical specimen. Media bottle. These bottles are suitable for storing non-hazardous chemicals or volatile organic substances. Eppendorf tube are used for preparing, mixing, centrifuge, transferring and storage of liquid and solid samples and also reagents. Test tube rack to hold multiple test tube upright at the same time. Sprit lamp used to heat up substance. Wire loop. The loop is used in cultivation of microbes on plate by transferring inoculum for sterilizing. Filter paper used for filter the samples. Multi-pipettes used to accurately measure and fill numerous vials of liquid at once. Microtitle well plate. Well plate are used in virology, serology, microbiology and myriad of other life science and drugs discovery laboratories. Elisa plate used for detecting and quantifying soluble substances such as peptides, proteins, antibodies and hormones. Thanks for watching. Please do like, share and subscribe.